All right, so we've got our next section here in the nervous system, and we're going to start by talking about, with this section, the central nervous system, and then from there move into the peripheral nervous system. And so for this class, obviously, we're not going to have a huge amount of time to get into all the different brain structures, but we're going to try to hit some of the high points and just kind of go from there. So, having nerves is one of those features that we associate with most animals. The only one that doesn't have nerves would be the periphery. Sponges, of course, don't have tissues of any kind, much less nerves or a nervous system. But once we got into the cnidaria, so jellyfish and corals and so forth, they're the ones that have the beginnings of a nervous system. At the very least, they have nerves that overlap in that nerve network that we talked about previously, but it's not a very dramatic nervous system. It's not super complex. There's no centralization. There's no brain. There's no spinal cord. There's no ganglia even. So it is a beginning of a nervous system, although a very simple nervous system. Right. So with that, cnidarians don't have any sort of associative activity. So not really much in the way of learning or anything like that. Although there is simple motor control with motor neurons and the ability to perceive the environment with sensory neurons, but that's really about it. With the flatworms, then, getting into the platyhelminthes, that was the first time when we really saw any sort of associative activity where they can, for example, if there is a dark part of the habitat and a light part of a habitat, they can move toward the dark part of the habitat. That's a very primitive type of associative activity that we associate with that group of animals. They're the ones that had the parallel nerve cords that run down the length of the body. Remember, a lot of the really primitive animals had more or less localized control in the body. So there was no centralized control where the brain was controlling what other parts of the body did. It was just each part of the body was essentially responsible for controlling itself. So muscle control, sensory perception, those kinds of things, but not a huge amount of associative activity. From there, as we got into the more and more complex animals, we started to see more and more complex nervous systems, and that's kind of the trend that we're going to continue here. Right, so getting into now vertebrate brains, if we sort of look at the broad classification of regions of the brain, broken down into three parts. There's a hindbrain, a midbrain, and a forebrain. Right, so the hindbrain is the rhombencephalon, and so we'll talk about some of the structures associated with hindbrain here shortly. But this is a really primitive part of the brain, and associated with a lot of our visceral functions, basic functional activities that take place in our body are coordinated by the hindbrain part of the brain. The midbrain is the mesencephalon, and so again, we'll talk about some of the parts there with the vertebrate brain, some of the structures associated with the midbrain, and then, although not too much in this course, also the forebrain, which is the main part of the brain that we're going to talk about for our purposes, that involves the cerebrum, the cerebral cortex, a lot of sort of higher level thinking with memories and language and in speech and those kinds of things will be centered more in the forebrain part of the brain. Now, in the fishes, which again are the most primitive of the vertebrates, their hindbrain is large. And so a lot of the hindbrain, basically think about this being more or less the brainstem where the spinal cord meets the brain. We have not only the spinal cord, but we have the medulla, which is responsible for a lot of very primitive but very important visceral function, and also the cerebellum, which is responsible for being able to perceive the body's position in space. And we'll talk more about the cerebellum here in a little bit. But basically, knowing where different parts of your body are in relation to not only the environment, but in relation to each other, is a really important function of the cerebellum that we see associated with the hindbrain. The midbrain is associated more with visual information, in fishes especially. In humans, the optic lobe of these cerebral cortex is very important for defining and coordinating visual activities as well. And then the forebrain, again, in fishes, is more in the realm of olfactory information. So if you're not familiar with olfaction, it just means sense of smell. Fishes are very big on chemoreception and detecting chemicals in the water. So they have an olfactory part of the brain that's responsible for processing that information coming in from chemoreceptors throughout the body. So if we look at the overall progression of the evolution of the brain in the vertebrates as a group, what we start to see is that as we move from the other chordates, the tunicates and lancelets, to the jawless fishes, which are the most primitive fishes, to cartilaginous fish, to bony fish, to eventually amphibians, reptiles, birds, and mammals, The one trend that you'll notice here, progressively, as we move up through the group consistently, is an increase in the size of the forebrain. So this purple region here starts to get bigger and bigger and bigger and bigger, but also more convoluted. So if we compare the size of the brain of, say, a duck and a cat, right? So roughly the same body size. Brains are roughly the same size as well, believe it or not, but the cat's brain has all these convolutions, all these wrinkles and folds, and all the things you sort of associate with a brain, kind of the wrinkly part of the brain. Um, that series of convolutions on the cerebral cortex provides a lot more surface area for more neurons to be able to fit. So even though the brains are the same volume, same size, the mammalian brain has a lot more surface area. And for that reason, it has a lot more neurons, and for that reason, it has a lot more processing power than that of, say, a bird. Now, birds have some relatively large brains compared to body size, but they're not quite as well developed as what ours are. So, going ahead and looking in more detail now at the forebrain, the diencephalon part of the forebrain, right? So this is still proencephalon, but the diencephalon part of the proencephalon, the back part of the forebrain, if you want to think of it that way, involves the thalamus and also the hypothalamus. So two really important parts of the forebrain. The thalamus is kind of like, I always sort of think about the thalamus as being the call center of the brain. So if you call into a big company, obviously you don't get to talk to a person right away. Eventually, maybe you do. But when you first call in, they want to know if you want to talk to sales, or if you want to talk to tech support, or if you want to talk to this or that. And you push buttons, and that eventually guides you to the right department. That's kind of what the thalamus is doing here in the brain. The thalamus is taking sensory information coming in from all over the body, whether it's pain, whether it's heat or cold or visual or olfactory or auditory or whatever, any sort of information coming in from sensory cells in the body. The thalamus determines where that information goes elsewhere in the brain. So it's like the call center that's routing information to the correct department and allowing it to be processed accordingly. Now, for individuals that have maybe an issue with the thalamus, maybe they're going to perceive a visual stimulus, but they're going to route that information to the wrong part of the brain, and they're going to perceive it as being a different type of stimulus. So maybe they smell baking bread, and they're going to perceive that as being a yellow triangle because the information was sent to the wrong part of the brain. The hypothalamus is a really unusual structure in that it's both a neurological structure as well as an endocrine structure. So the hypothalamus, first off, is involved in maintaining a lot of homeostatic function in our bodies. So body temperature, for example, that particular trait is maintained in our body via the hypothalamus. Hypothalamus is also part of the limbic system, which we talked about previously when we were discussing cocaine and how that affects the body. The hypothalamus is also a big, big driver of the endocrine system, and it actually produces some of its own hormones, but also by producing those hormones tells the other pituitary glands what to produce and when to produce it. The telencephalon is the end brain, and this is really the main, most complex, most human-associated part of the brain. This is where we find a lot of our associative activity. And again, when I say associative activity, I'm talking about higher mental functions, memory, language, speech, abstract thought, those kinds of things are all part of the end brain. And so in our case as mammals, that's going to be in our cerebral cortex, which we'll talk about more here in a little bit. The cerebral cortex is divided up into two sides. So there's a right hemisphere and a left hemisphere. They are fairly symmetrical. They look very, very, very similar on both sides of the brain, but there are differences in how these parts of the brain perceive the environment. The right side and left side of the brain, first off, are connected by a layer of tissue called the corpus callosum. Now, this tissue is what allows for the right side and left side of the brain to coordinate. And so if this corpus callosum were to be severed, or if the right side and left side were to be isolated from each other by a loss of connection through the corpus callosum, then the right side and left side of the brain actually lose a lot of coordination. So what's happening on the right side of the body in that case would actually be largely independent from what's happening on the other side of the body, if the two sides of the brain are not able to communicate anymore. Something else that I think most of us are kind of generally aware of is that each side of the brain is responsible for processing information from the other side of the body. So, for example, the right side of the brain receives sensory information coming in from the left side of the body, and vice versa. The left side of the brain receives information coming in from the right side of the body. So, a little bit unusual, and we have this crossing over, or it gets a more technical term is decussation of the information coming in from one side of the body to the other side of the brain. Crossing over is perfectly fine. We don't need to worry about decussation, but that is something that happens within the brains. Now, on each side of the brain, each hemisphere of the brain, there is also then a lobe. So there is a frontal, a parietal, a temporal, and an occipital lobe going from front to back or anterior to posterior on both the right side and left side of the brain. So left frontal, right parietal, right parietal, and so on. Each one of these lobes are responsible for different jobs. All right, we'll talk more about those here shortly. Now, again, the cerebral cortex is a bit unusual in mammals because we have this very, very highly convoluted nature where the cerebral cortex has these wrinkles and folds. Instead of just being a smooth surface like we would see in like a bird or a reptile, this convoluted wrinkled surface gives our brains as mammals a lot more surface area and therefore a lot more potential to have more neurons packed into a smaller space. And having more neurons essentially translates to more processing power. The more nervous cells you have, the more information you can process, the faster you can process it, the more things you can do. So the cortex then is like we said, divided up into regions. We also have these lobes here. This is a really good picture because it shows us a lot of the big kind of surface features of the left side cerebral cortex, where we have, again, frontal, parietal, temporal down here, and occipital in the back. And then within that, we also have areas, right? So we have Broca's area up here in the frontal lobe, which is responsible for speech. We have Wernicke's area up here in the parietal lobe, which is responsible for interpreting information coming from the outside world. So one of the things that we're going to see is that language is actually a function of our brain that's spread throughout a lot of the cerebral cortex, because language isn't just one thing, right? We can understand language that's being spoken to us. We can understand language that's written down on a piece of paper. We can produce our own language either by using the muscles in our hands to write or using our vocal cords to speak. And so all of these different aspects of language are coordinated by different parts of the brain, which is kind of interesting, especially when somebody experiences a damage to the brain where maybe one of those areas is damaged but the others are okay. They might lose the ability, for example, to perceive written speech, but they might be perfectly capable of producing their own speech or understanding spoken speech, even though writing or understanding written speech would not be something they could do anymore. Within the brain then, the three areas that I was talking about prior to this was the primary motor cortex, the primary somatosensory cortex, and then we also have our associative cortex, right? So the primary motor cortex is the part of the brain associated with controlling movement, voluntary movement in this case, right? So this image here, which is always kind of interesting, it's very sort of bizarre looking at the features on the sides of this image. But what this is trying to show us is sort of like the relative amount of neurons devoted to each of several different parts of the body. So if we go over here and look at the primary motor cortex, by looking at what's called a homunculus in this case, and homunculus goes back to centuries ago. It was a term that was used to try to understand how people developed before they were born. They actually thought back in the day that when you were developing in utero, there was actually a very, very small, tiny, fully formed human that was present inside the uterus that would eventually emerge as an infant. Now, it's not really how it works, but that was kind of the idea, and that was how this particular image, this sort of map of the brain was named. So anyway, back over here, the primary motor cortex, the idea here is that the relative size of the body part in this picture sort of is an indication of how many motor neurons we have associated with that body part, right? So the larger the body part is, the more motor neurons we have proportionally going to that part of the body. So if we look at, for example, the hands, right? The hands have a lot of very, very fine motor control, a lot of small motor units. We need to have a lot of motor neurons there to provide us with the dexterity that we need to actually control our fingers and our thumbs and our hands as a whole. So we have a lot of motor control there. On the other hand, if we look at, if we look at the elbow, right? The elbow doesn't really have a whole lot of motor control because the elbow isn't really, it's just a joint, right? The muscles that control the elbow are going to have a lot of motor control, but the elbow itself doesn't. So it's not going to have as big of an impact on this picture. Now, if we look at the primary somatosensory cortex, kind of the same idea, right? So the size of the structure over here is an indication of how many sensory neurons we have associated with that particular part of the body. Over here we have the hands, again, lots of sensory neurons because tactile sensation with our hands is kind of a big thing that we use. Lips and teeth also have a lot of sensory neurons. If you've ever had a toothache or something like that, you know that that's definitely the case. And other parts of the body don't have, again, as much sensory control. Elbows, again, not really known for being that sensitive, so they're not going to have that many sensory neurons. And then the association cortex is where, again, we find our mental functions. So that's where we find our learning and memories and so forth centered in the association cortex. Within the brain, we also have what are called basal ganglia. And so ganglia is a term that we haven't really talked about too much yet in this chapter, although we did cover it in previous chapters. A ganglion is a collection of cell bodies. So a bunch of neurons, specifically the cell bodies of neurons, clustered in one location, in this case in the brain. Basal ganglia are there, and they are going to help control different parts of the body. And again, this is somatic control. This is voluntary control, moving different parts of the body. We talked about the thalamus already and the hypothalamus, so those are two structures we've kind of already seen. The limbic system as a whole is part of the body's what's called pleasure pathways again. So the hypothalamus, the hippocampus, and the amygdala collectively make up the limbic system, which is kind of like a subsystem that's comprising part of the brain. The limbic system in general is also responsible for a lot of emotional responses, especially very primitive emotions like anger and fear and sexual arousal and those kinds of things. Those emotions are very much associated with the limbic system. We can also look at the arousal of the brain in sleep. The reticular activating system is a subset of brain structures that are going to allow us to maintain alertness and consciousness, and when they're not as active, they're going to allow us to go to sleep. We can monitor this with an EEG, which is sort of just a way to measure the electrical activity of different parts of the brain. The idea with the reticular activating system is that the more stimuli coming into the brain, the more active the RAS is going to be, and the less likely you are to go to sleep. So if you're in a room where there's a lot of movement and light and activity and sound and just a lot of just busyness going on, the reticular activating system is going to be more aroused, and therefore you're going to be less likely to be able to fall asleep. If you're in a dark room where it's quiet, you're going to be more likely to go to sleep because the RAS is going to be less stimulated and therefore allow you to fall asleep more easily. As far as, again, the two hemispheres, and looking more specifically here at language, the left side of the brain, left hemisphere, is the dominant hemisphere in most individuals. So this is where we're going to find a lot of the language centers of the brain located in the left hemisphere. The left side of the brain is very, very good at working with sequential reasoning, which kind of sort of makes sense because if you think about it, language is very much reliant on sequential kinds of things. You've got to put words in order, you've got to put letters in order, sentences in order. It's very much a sequential sort of a thing. So the left side of the brain is kind of responsible for that aspect of language. The right hemisphere is more of the spatial reasoning side of the brain. So we see a lot of artistic ability, a lot of musical ability, also a lot in the way of memory formation and sort of remembering nonverbal sorts of experiences as well that are kind of like back there in the memory banks maybe we don't think about every day. A lot of that is also related to the right hemisphere of the brain. Now, speaking of memory, this is something that is very relatively poorly understood in terms of neurology. Short-term memories, which are generally going to consist of things that are sort of retained in the brain for maybe a second or two. Short-term memories are described as being transient neural excitations. So if we kind of break that term down, transient means they're temporary. They're there and then they're gone eventually, fairly quickly. And most of these short-term memories will never be recorded as long-term memories. They're never going to be as permanent as a long-term memory, which is actually a really important thing. There have been individuals who are able to retain most of their short-term memories as long-term memories, and although that might sound like it's a great beneficial thing to have because you can like never forget anything and it's really easy to study and take a test and remember someone's birthday or whatever, it's actually very disruptive because most of those short-term memories that we experience, that we possess, are not really important enough for us to hang on to. And if you simply remember everything that you're exposed to, the really important stuff gets kind of lost in the shuffle, right? So if you remember every little aspect of your day, if you remember exactly what you had for breakfast back on March 19th of 2008, that kind of level of remembering details would actually be extremely disruptive to your functioning as a normal individual. So it's actually fairly important that we do forget most of the short-term memories and only hold on to the significant long-term memories over longer periods of time. Long-term memories actually involve physical changes to the brain itself. They are sort of rewiring of the brain where you've got connections forming among neurons or breaking between neurons. They involve physical changes to the brain itself. So the difference then between a short-term and a long-term memory is actually, it's pretty substantial. The short-term memory is just a really short burst of neural activity, and the long-term memory involves an actual physical change to the brain itself. How do we go from short-term to long-term memory? We don't really know. We don't really know, first off, how memories are chosen to be stored as long-term versus those that are lost as short-term memories. And we don't really understand the process of turning a short-term memory into that long-term memory, which involves a change in the physical structure of the brain itself. We think the hippocampus and amygdala are involved in some of this conversion. Part of the reason for that is that a lot of other animals, birds, for example, a lot of times will have a very, very large hippocampus, and those birds are very, very good at remembering things. So you might find a species of nutcracker out west in the U.S. that can store 10,000 seeds in the fall and literally remember where every single one of those 10,000 seeds is in its environment. That requires a very, very good degree of long-term memory that most of us probably don't have. But that species of bird also has a hippocampus, which is actually, I think, the biggest part of its brain is its hippocampus, which is a bit unusual, but it does allow it to remember over the long term. Another kind of feature that comes up when we talk about memory is Alzheimer's disease, where this is a very broad category of things, but essentially what happens is that the memory process is disrupted. The thought process is disrupted. So a lot of what we think happens here is that conversion process of short-term to long-term memory is broken. A lot of times people that have Alzheimer's can still remember long-term things that happened maybe decades ago, but they can't remember what they had for breakfast that morning, that kind of a thing. So it's a very extremely disruptive situation that occurs, and unfortunately most of us have known somebody or will know somebody that suffers from this, but it's a very, very dramatic and kind of a tragic thing that occurs when somebody, especially if they're very healthy otherwise, if their brain is degenerating as a result of Alzheimer's, it's very difficult to watch that proceed. In terms of what causes Alzheimer's disease, and the reason why I have this is actually a picture of the dorsal side of a shark, so we can see where its eyes are, we can see where most of its brain is, right up through here. The reason why this is, I showed this in the slide, is because there's a lot of interest in sharks because they are, some of them, fairly long-lived species, but for whatever reason, their brains do not seem to suffer from the same kinds of degeneration that ours do when it comes to Alzheimer's disease. And so there's been a lot of interest in trying to study them to figure out what exactly about their brains makes them resistant to the neural degeneration that we associate with Alzheimer's. One hypothesis that actually has a lot of support for it is that there is a type of protein produced by cells in the brain called a beta amyloid protein. It's basically a waste product that occurs as a result of nerve cells functioning in the brain. And if this beta amyloid protein is not removed from the brain, if the waste is not disposed of properly, then it begins to build up and actually more or less smother the cells in the brain, some of the cells in the brain, thereby causing them to die off. And that will then, of course, lead to long-term degeneration of the brain. The brain does have its own lymphatic system, which is something we've not known for very long, maybe only a few years, 5 or 6 years at most, that functions primarily when we're asleep. So one of the really important functions of sleep is to allow the brain's lymphatic system to sort of rinse out some of these structures of the brain and remove, among other things, some of these beta amyloid proteins that can build up and become toxic to the brain itself. There's another hypothesis where there are proteins produced inside the cells that cause their own death. These are called tau proteins, which this one actually has a bit less support, but it definitely is still on the table as being a possible contributing cause to Alzheimer's disease. There's a lot of other interesting things here as well. Some of these beta amyloid proteins especially can serve as prions. So we've talked about those before, I know in Principles 1. They can cause other proteins around them to become beta amyloids as well. They have a prion-like function, which can be, of course, it can accelerate the rate of damage of these proteins on the brain itself and accelerate that degeneration. So we're going to stop here on this section, and when I pick back up, we are going to do one more section on the nervous system, and then we'll wrap up the chapter from there.